Hello, everyone, and welcome to today's webinar. My name is Olga Bodie, and I'm part of the team here at SYNGAP Research Fund. Our presentation today is Behavior, Cognition, and Sensory Processing in People with SYNGAP1. And I have the pleasure to introduce today's speaker, Dr. Andrew Stanfield from the University of Edinburgh. Dr. Stanfield is a senior clinical research fellow at the University of Edinburgh's Division of Psychiatry and an honorary consultant in the psychiatry of learning disabilities, working in hospitals in Edinburgh. He's also co-director of the Patrick Weil Center for Research into Autism, Fragile X Syndrome, and Intellectual Disabilities. Dr. Stanfield's research interests primarily relate to autism spectrum, intellectual disability, and related neurodevelopmental disorders. Recorded version of this webinar will be available on our SRF website under the resource tab. And by the end of this presentation, you'll have the opportunity to get the answer to your questions, and we'd love to hear from you. So please put those questions in the Q&A below. And for those of you just joining us, welcome. And again, our speaker today is Dr. Andrew Stanfield from the University of Edinburgh to discuss behavior, cognition, and sensory processing in people with SYNGAP1. It's now my pleasure to turn things over to Dr. Stanfield. Thank you.
Okay, thank you very much, Olga. I hope you can all see and hear me fine and can see the slides. Do let me know if you can't. So it's a pleasure to be speaking to you. I'm going to start off with an apology. Well, two apologies actually, to be honest. The first is that I currently have COVID. So if I appear to be a bit spaced out, or maybe more spaced out than usual, that's my reason. The second is that there's quite a lot of data and things in this presentation, because what I'm going to do is present you some findings from research that we've been conducting over the past few years, and which indeed I think if there's some of the UK families are watching here, which I'm sure there are, then you will certainly recognize some of the questionnaires and some of the tests and things I'm going to present. Before I get on with the actual presentation, just so that people know up front, me and my research group have received funding from various pharmaceutical companies for clinical trials, and also for doing consultancy over the last 10 to 15 years, and particularly for trials of autism and another condition we work in called Fragile X syndrome. And I work at a place called the Patrick Weil Center at the University of Edinburgh. And as part of that, we work alongside, or really with, another centre called the Symes Initiative for the Developing Brain. We describe these as being sister centres at the university. And what they do, our whole kind of idea, and this is taken from the SIDB website, is that we're trying to understand the brain basis of, and to test new therapies for really single gene conditions which are associated with autism and intellectual disability. And you can see from, as I say, this is taken from the SIDB website, SYNGAP1 is right there as one of the big genes that we want to understand, and the big conditions that we are working on here in Edinburgh. The work that I present today is one aspect of that. But these are all the different group leaders from SIDB. And each of these individuals works some to a greater and some to a lesser extent on various genes associated with neurodevelopmental conditions. And each of them runs a research group. So I suppose I'm just trying to get over to you that there's a lot of people who are working on this field here in Edinburgh, a lot of them based in sort of laboratory scientists, and then some clinicians like myself. So I'm going to present kind of one aspect of the SYNGAP1 program in Edinburgh, but there are lots of people here who are working on SYNGAP1. I mean, you can see at the top, Peter Kind is the head of SIDB, who's done a lot in SYNGAP1 over the years, and many of the other people here, some of whom may have indeed spoken to you before. The other thing about the centres I want to get across before I start is that they are, they were started by families, really. That's been at the core of what we do since the beginning. The Patrick Weil Center started now, something like 13, 14 years ago. And it was this family, the Wild family here. This is Alfred Wild, who left money and a legacy donation really to set up the Patrick Wild Centre, named after his brother Patrick, who had severe intellectual disabilities and autism, but actually more so in memory of his parents who cared for Patrick at a time when there was little or no understanding or support available. Other families have been involved, and some of whom are pictured here throughout. And then in the bottom, this is from one of our family, the Syngap family meetup in the UK from last year, which weirdly I also had COVID for. So I had to talk online at that. But we have another one coming in a couple of weeks' time. So I'm hoping, well, I will definitely be well enough to attend that. Okay, but what I'm presenting today, or what I'm going to present, is findings from the NeuroGD study, which is really a large study, and it's SYNGAP1 is one of the main genes or one of the main conditions that we're looking at in this, and we were also looking at another one called Fragile X syndrome, but obviously I'm going to present mainly the SYNGAP results today. And let's say some of you may recognize some of these tests. Damien and Aisling here were the two people who did a lot of the work, traveled around the UK. Some families came to us, some we went to see in other parts of the UK to take part in this study. And what we were aiming to do in NeuroGD was this. We were trying to really understand behavior and to describe behavior in detail in people with SYNGAP1, and to describe a bit about cognition, a bit about sort of how people think and how people reason and work things out in their brain. We were looking at whether or not we could do research level EEGs in people with SYNGAP1, and I'll present a little bit of that data that shows that we did. And we were looking at whether or not we could use some of these measures, things like the EEG, things like the cognition, whether or not they might be suitable for future trials to think about tracking response to treatment objectively. So rather than just using measures of behavior, which are inherently subjective, looking at these other sort of more objective measures to see about whether we could track sort of progress and response to treatment. And then we were also interested in basically establishing cohorts, getting in contact with people, getting to know people, with the idea that at some point, what we aim to do are trials in SYNGAP1, as we have done in other conditions. So that's one of our big aims, is just around sort of developing those cohorts, developing those contacts and things through all of this. So when you're interested in behavior, what we decided to do first, and I have to thank Mick Parker for this, who's one of the geneticists who did some of the early studies in SYNGAP1 in the UK. Mick said, well, the first thing you should probably do is just go and ask the families. And I think that's maybe something that we don't do quite enough. So we did. We did some of these clinical interviews with families. We started them at our first family meetup. And we then, we're basically looking for themes, of when we were asking people about, how are your children, and we're mainly focusing on challenges, I have to say. I think that's a gap in what we do, generally speaking. But we were mainly focusing on the kind of behavioral challenges that people are faced with. And so we did these interviews in about 27 people, mainly children. We were asking families about how they were. And you can see the kind of themes that came up will not be a surprise, I guess, to any of you. But these sort of repetitive behaviors, difficulties with change, people struggling with transition, getting very focused on things. Combining that with some of the social difficulties and things here these are kind of classic sort of autistic-like traits. Often associated, and I'll talk about this in more detail, things like distress-related behaviors, becoming aggressive towards themselves and other people. These sort of, people sometimes call them challenging behaviors. Were quite commonly brought out. And then struggling with daily living skills, struggling with emotional regulation. And then I'll talk more about sensory differences later on, but it was one of those things that came up quite commonly. And people very concerned about this lack of danger awareness, being unaware of danger, sort of not really being able to pick out, run into the road, not knowing kind of the general life or social dangers that might be. So this just gives us a bit of a handle on which to start, to think a bit about what more we want to try and measure, what we want to get into in a bit more detail. And we were doing that through these kind of more quantitative measures of behavior. So things that we can actually, count and allocate scores to. And this is very common in this field. I'm sure most of you are very familiar with it. Looking at things like, we did a whole load of questionnaires. Anyone who took part in this study will remember probably doing a whole load of questionnaires. But we looked first of all at this adaptive behavior. So the kind of things that people need for day-to-day life. We used various scales, the thing called the Vineland for that. We looked at, I'll talk more about these scales in a bit. We looked more into these behavioral and emotional difficulties. We looked at autistic traits and ADHD traits, and we looked at sensory issues as well. So I'll show you some of the findings and some of the results from these. Oh yeah, we did this in a group, similar kind of group, same sort of group. These were all recruited, or mainly recruited from the UK, not entirely, but mainly recruited from the UK. About 30 people, 30 with SYNGAP and just slightly less. TDC means typically developing controls. That's like a comparison group. We had a few adults. Almost all of the data I'm going to present, though, relates to the children, because we didn't really have enough adults to make great sense. I think there was only 3 people above the age of 18 who took part in the study. So everything I'm going to present here is going to be about children. Of the 30 people with SYNGAP, about 40% had a diagnosis of autism. That means that they'd actually been seen by somebody and been formally given a diagnosis of autism. Only 2 had a diagnosis of ADHD, which is important, and again, I'll come back to. And about 60% had epilepsy, which I was surprised at. I thought that would be higher, to be honest, given that a lot of the SYNGAP research has come out of the epilepsy field. And then the mean kind of non-verbal IQ was about 60. Again, I'll come back to talk about some of these things in the future. So starting off with the adaptive behavior on the Vineland, this gives you can get all kinds of scores from these things. Indeed, one of the difficulties is sometimes trying to reduce the amount that you're actually measuring, so that you can make best sense of it. But so basically what the Vineland does, it gets people to answer loads of questions, and then it gives you scores for different domains. Communication, which consists of 3 subdomains: daily living skills, socialization, and then motor skills, so fine motor and gross motor for that. Importantly about this, the scores are all age normalized to a mean of 100, like a bit like IQ. So, the level of communication a 4-year-old has to show to get a score of 100 is much lower than the level of communication a 15-year-old would have to show. When you expect a 15-year-old to do more in terms of daily living skills, looking after themselves, getting out and about in the community, than you would do for, say, a 3-year-old. So they're age normalized, and that's important for what I'm about to present. Lots of graphs, lots of data coming up. I will try and just explain the important points. Basically, the key takeaway here, which again is not a surprise, is that in our SYNGAP group we see difficulties really across the board in terms of daily living skills and adaptive kind of behaviors. SYNGAP, the SYNGAP group will be in red in most of the slides that I present. What's I guess important, is that I think the most marked differences are seen down in communication. So, even compared to the other scores, the children show kind of receptive and expressive communication difficulties, which are probably more pronounced than one would expect, given some of the other difficulties that motor was a bit less spared than the others, but still, there's still quite a lot of motor difficulties that people have, and that we know about. One of the interesting things that comes out from this is that the older people in our cohort have got lower age normalized scores than the typically developing folk. Now, what that means, I think this is important, is that this is, as I talked about, well, 2 things. One, this is cross-sectional data. So this person here who's about 12, 13, 14 years old is different from this person here who is 4 years old. The way you want to look at this to show it's actually true is to look at it longitudinally. So to see this person here at 4 years old, and then to track them up to 8 years old, 10 years old, to see how these scores change. So first of all, it's cross-sectional. So it may just be that some of the older children have got more difficulties, and that could just be coincidence. The second thing that's important about this, these are kind of age normalized scores, as I say. That doesn't mean that. So what I'm not saying that people are declining per se. This isn't talking about a regression or that children have get worsened or lose skills. What it is more is that some of these more complex skills, particularly things like learning how to write, going out and about in the community. You see that these are non-age normalized scores, these ones I should say now. In the controls, you see these pick up dramatically between the ages of 4 and 14, and that's again to be expected. And that's not what you see. You do see improvements, you do see development in our SYNGAP cohort, but it's not to the same extent that you see in kind of controls. And it's in particularly these kind of advanced skills that people would normally pick up as they get older. The other thing that was interesting about this is that it was most marked, this kind of not keeping up with skills or not developing things, in those with epilepsy as well. Now whether or not that holds whenever we look longitudinally, I don't know. And what that means is also something that we don't know. It's possible that some of the older children were more likely to be epileptic, and that's how they got diagnosed 10 years ago with SYNGAP1, and so that's why this relationship is there. It is also possible that people who have untreated or lots of seizures and things, it would be understandable if maybe they didn't manage to develop the same kind of skills as those people whose seizures are maybe kind of well controlled. So this is stuff that we need to look more into longitudinally. The next thing that we looked at were these kind of behavioral difficulties. And this was using a thing called the CBCL, awful questionnaire, 113 questions, quite small writing. It's a very good questionnaire, lots of detailed information, but just when you're the person filling it in, it can take a long time. It covers lots of different domains. It looks at kind of what are called internalizing problems. That's things like withdrawing into yourself, being anxious or depressed, sort of somatic complaints or physical health, aches and pains and things like that. It looks at externalizing problems, things like aggression and rule breaking, are the kind of important parts of it. And I think of things often that cause the most difficulty. And then it also looks at things like attention and social difficulties and difficulties with people's thinking and processing of information, which are associated with intellectual disability, autism, and ADHD. And if you look at this in this sort of set of scoring, the controls again are in blue, and we see particularly high levels. I was surprised actually. I thought we would score higher levels on things like anxiety and things in the SYNGAP group, and I wonder if maybe we're just not capturing that well. But we do see quite high levels of attentional problems. Particularly interesting, and I'll come back to this later, lots of our group do not have diagnoses of ADHD, and high levels of aggressive kind of behaviors. These social difficulties associated with autism and the thinking and thought difficulties are most likely related to ADHD, or not to ADHD, to intellectual disability. But that holds when we look down at the kind of, if you look at this scale, at the items that were endorsed by over 50% of our population, lots coming out in the attention hyperactivity, and quite a lot coming out in aggression and agitation sides of things. That these are, relatively common within this cohort and cause difficulties and stress associated. Again, we wanted to look at age, because when we speak to families, there are some families who've come to us and said that that they perceive that things have been getting more difficult through adolescence, and certainly within our childhood cohort, we do see some of these difficult behaviors become a bit more marked over the course of adolescence. Again, this is cross-sectional behavior, cross-sectional data. So we need to look at this longitudinally to see whether this actually holds. The next set of things that we looked at were autistic traits and ADHD kind of traits in terms of behavior. And you know, what we find in terms of autism, so this is a thing called the social responsiveness scale. And we find really quite high levels of autistic traits according to this scale in SYNGAP1. About 80% of people are scoring in the moderate and above impairment on this scale. Now, this is not a diagnostic scale. This is a screening scale for autism. What's interesting about it, doesn't relate to age. So this is not something that changes with age, which fits with autism, that the traits really aren't supposed to change. It is very much associated, though, with nonverbal IQ. And it's possible that some of these autistic traits might actually relate more to the difficulties, the broader difficulties with intellectual disability and so forth, rather than autism per se. What is interesting, though, is that about 40% of our population had actually a diagnosis of autism. So, you know, that is either underdiagnosis, you know, that actually there are more people with autism who aren't, for whatever reason, getting a diagnosis. And there's lots of things that affect whether you get an autism diagnosis. Certainly in the UK, there's very high waiting lists for some of these things. It depends who you're seeing. You know, these things, autism diagnoses might be more likely to be made in certain disciplines and so forth. And it depends whether or not, you know, it's been felt, you know, some doctors will say, well, you know, what's the point of giving an extra diagnosis? We know that this is related to this genetic change. So, you know, there's lots of things that affect it. But it is possible that people are being kind of underdiagnosed or the autism being under recognized. It is also possible, though, that at least some of these traits are more related to the sort of more global intellectual difficulties as opposed necessarily to autism per se. What is important is when we look at whether or not autistic traits, how they might actually relate to these measures we looked at earlier, things like adaptive behavior. The more autistic somebody is in terms of their social communication, the lower their adaptive behavior is. So that means that people, generally speaking, more autistic people are going to struggle more with the kind of things that allow you to get out and about in the community, to manage yourself, to communicate with others, and to do your kind of daily living skills and things. What's interesting is that social communication skills, autistic, aren't really related to externalizing behavior. So these kind of aggression, these behavioral challenges, there's a slight relationship here. It's not statistically significant, but it's not a big driver of these kind of behaviors. For repetitive behaviors, we see it's slightly different picture. You know, the levels of repetitive behavior, and this is things like resistance to change, problems with routine, problems with transitions, relates a little bit to adaptive behavior, you know, but it's not super strong. But it does relate to these externalizing behaviors, to aggression and so forth. But these sort of things like resistance to change and repetitivity, you know, are associated with higher levels of aggression, higher levels of you know, frustration, lashing out and things at other people. And I do wonder if I mentioned earlier on that, you know, we didn't score particularly highly on levels of anxiety in some of our groups using the scales we looked at. But I suspect that at least some of these sort of things, like the resistance to change and struggling with routine, these are anxiety related, or these can cause a lot of anxiety and agitation. Children who struggle with changes to routine can become distressed and stressed, maybe rather than anxiety, distressed, more often, and that can then lead to these kind of externalizing behaviors, the things like aggression and so forth. ADHD traits were slightly different. I was particularly interested in this because I was surprised that, you know, I don't know if I was surprised, the relatively low rates of ADHD diagnosis in this sample. The 7%, only 2 people. Now, this relates possibly to 2 things. One, this was mainly a UK sample. And in the UK, we do diagnose ADHD less than in some other parts of the world, particularly the US, although it's not that much less than it used to be. The second is that I think, broadly speaking, across intellectual disability, we underdiagnose ADHD. There's a tendency to attribute things like inattention, hyperactivity, to IQ, it's part of a global impairment, these global IQ differences. Whereas actually some of it, might actually be a similar process to what leads to ADHD in the general population. If you've got a general population rates of ADHD, which can be between 5 to 10%, depending on where you measure them, there's no reason why you wouldn't see that within our SYNGAP populations, within our learning disability populations, and we're likely to see it more. So the ADHD was particularly interesting. It didn't relate to age, so it doesn't change with age, and it didn't relate to non-verbal IQ. So it's not a bit different from the autistic traits. This is not in this cohort anyway. It's not the people who have got lower IQ have more ADHD. It seems to be something separate. Okay. So in terms of ADHD traits, the other thing that's really interesting about them is that they're a bit associated with adaptive behavior. Kids who don't pay attention as much, do struggle a bit more with adaptive behavior, do struggle a bit more with behavioral difficulties. But the hyperactivity, impulsivity side of things, part of the ADHD. Hyperactivity, impulsivity part of the ADHD is very much associated with difficulties with adaptive behavior and very much associated with difficulties with externalizing behavior. Okay, I should say, sorry, I can hear at times that there's various noises going up, and I'm not quite sure whether those are related to what's going on in the webinar. I'm unable to see the chat and things at the moment, so if there is questions and things, we can come back to them, or someone's able to I'm happy to be interrupted if they wish to. But yeah, particularly this hyperactivity impulsivity component of ADHD seems particularly important to determining how well people get on with their adaptive behavior and the levels of behavioral difficulties that they show. So lots of graphs, lots of data. There'll be more to come, I'm afraid, but going back, just trying to summarize some of these behavioral findings that we have high levels of autistic traits and high levels of ADHD traits across all age groups. Despite, diagnostic rates of these conditions are relatively, 7% for ADHD, 40% for autism, maybe underdiagnosed. I think particularly important to consider this for ADHD because ADHD might, there are treatments for ADHD. They probably don't work quite as well in folks with intellectual disabilities and that, attentional and hyperactivity in the kind of more global cognitive impairment sense. But there are treatments and they are things that could be tried and that could have some, significant benefits for people. I think it's important to point out, I think everybody here knows communication is a particular problem and that is important. And I think, having access to speech and language therapy, to look to develop functional communication skills. I'm not saying necessarily people will develop lots of language, but better ways of communicating and supporting communication are really important. I think that thinking about the behavioral difficulties, which is obviously one of my main interests being a psychiatrist, focusing on anxiety, the kind of things that maybe drive some of these difficulties with routine and so forth, and focusing on ADHD would seem particularly important if you're looking at trying to treat or manage some of the behavioral difficulties that occur. And then the last thing, I'm very tentative about saying this because, as I say, this was cross-sectional data. There's some idea that some of our older adolescents are maybe more affected than the younger, or the ones in our cohort were. But whether this holds true for, as we study people over time, whether we see changes in these difficulties, we need to work out. So we need to do longitudinal studies to see if this is true, and then to look at the reasons as to why that might be and what might be done to help. Okay. That's most of the stuff from behavioral questionnaires. I'm going to very briefly mention sleep. This is not work that I have done particularly. I was, although we did data from NeuroGD, we contributed to this paper with Connie, Constance Smith-Hicks, and Jimmy Holder, who will be familiar to many of you, looking at sleep, because sleep is obviously one of those things that families, struggle a lot with, and it can be particularly difficult. And we really, what we can see is that, it's like, as families have said to us, children with SYNGAP1 do struggle to sleep, and they're actually probably more severe sleep difficulties than are seen in some other forms of genetic intellectual disability. So this paper was comparing SYNGAP1 to Phelan-McDermid syndrome, which is another genetic form of intellectual disability in autism, and found that the SYNGAP kids, had similar or greater difficulties with sleep. Colleagues of mine, Lindsay Mizzen, again, will probably be known to some of the UK families, went out and did some sleep studies using actigraphy watches, and did some sleep studies where they actually went to people's houses and attached various electrodes to measure brain waves and things like that to people, to try and get a handle on what these sleep difficulties are and what they might be caused by, or what the patterns are. Lindsay, I'm sure, would be delighted to come and talk about this with you at some point. This some of her early findings that she's looked at and has presented elsewhere, showing that, we can measure, we can use this actigraphy, these watches that kids manage to wear, to actually quantify the sleep difficulties, to actually measure how much people are awake after they go to sleep, and then how efficient their sleep is. So if you spend time in bed versus time asleep, basically, is sleep efficiency. And this is important because if you're doing studies of medicines, either to help sleep or to help other aspects of SYNGAP, using these actigraph watches could be particularly useful. The other thing that Lindsay is doing is looking at some of the findings from the more detailed sleep studies and comparing those to findings that colleagues of ours in Patrick Wild Centre and in SIDB have in the laboratory models of SYNGAP, to look at these kind of translational biomarkers, as they are called. So I won't go into that in any more detail, but it's just to let people know. Okay, where am I for time? Cognition, then. We wanted to also measure cognition. A lot of the behavior things we had looked, we had some ideas of the kind of things that we might see from talking to parents, but we wanted to try and understand, how thinking was affected in SYNGAP1. And this is quite difficult to do, because one of the main ways that we access people's thoughts is through what they say. We wanted to be able to understand how the brain works, how people think, and how that might be different. We also, as I said earlier on, we wanted to look at more objective ways of capturing difficulties. I've talked about social difficulties, attentional difficulties, and so forth, but we wanted to look at whether or not we had ways that we could measure these that were not just, rating scales, which are affected by how things have been that week, what's going on in the child's life. They're affected by how you feel, they're affected by how the child feels, and they're affected by actually what the investigator says to you. Whereas what we wanted to do in measuring cognition was to try and tap something a bit more objective, using these measures. We used a non-verbal IQ measure, which I've already referred to a little bit, called the Lighter 3. And then we used measures of social attention and inspection time, which is a kind of more global cognitive measure, using a thing called eye tracking, and I'll talk about each of these. So the Lighter 3, first of all, I don't have a huge amount to say about this. This is basically a non-verbal IQ test. Again, it's age normed to about 100 to 100. So, a child who is 3 or a child who's 15 has to perform better than a child who is 3 on it. And it is entirely non-verbal. So it's not, the idea of using this was to try and get at, the population of children that we saw, how, what their IQ was if you took away some of the communication. Now, I don't think it's possible to entirely divorce findings from communication. There is a level of communication even in administering a non-verbal IQ test, but it's just, it probably represents a bit more people's underlying cognitive abilities. So most IQ tests, I should say, I guess people might know this, are a combination of verbal IQ and non-verbal IQ. And one of the concerns really is that, when you do that, if you have a population who particularly struggle with communication, as the SYNGAP1 group do, your score that you're getting in an IQ test maybe doesn't reflect someone's actual abilities and understanding and ability to think and reason. And so whereas non-verbal IQ might be a better measure of how people actually function. And then what was interesting to me in this was the spread, really, more than anything else. You know, the mean IQ was in the non-verbal IQ was above 60. It was just above 60. That's higher than I expected, to be quite frank. You know, and it stretched up. We had one person with an IQ of 100, IQ of 90, down to 80. I mean, you know, and they did go down to the lower end of the range, but it was the spread of IQs, and to be honest, I think higher IQs than we expected, which I think is probably, I mean, you may well, as parents, may well know this, that people seem to understand maybe or pick up more than maybe their levels of communication actually allow them to demonstrate. And I suspect that's what we're tapping here. And we can see that nonverbal IQ is important. I mean, it is, it does relate to people's adaptive behavior. Those people who score more highly on this IQ measure tend to have a higher level of adaptive behavior than those who score less well. So, you know, I think it's just interesting, and it speaks to the idea that actually, you know, that there are perhaps skills and abilities that children just maybe aren't able to show because of their levels of communication. Eye tracking we used to look at particular, as I say, this inspection time and social intention. The reason for using eye tracking, I mean, basically what it is you have a computer, and sat at the bottom of this screen here is a little device that tracks where people look at. And you can use that to measure various cognitive abilities, because you can look at where people are looking at, and you can look at how long people are looking at it for. And it's passive. The nice thing about it is it doesn't require communication. It doesn't require, you're not telling the children, okay, you know, look at this screen and find the O, or look at this screen and do this. It is literally, you're showing them the screen and you can see where they look at. And because it's passive, it's quite suitable for non-verbal groups. And then because it's objective, we're not telling people to do things, and we're not asking people to measure things. We're just measuring what is actually a behavior that's occurring. It's potentially useful as a measure, you know, to tap this cognitive thing as a measure of possibly response prediction to medicines and things in trials, possibly for use as an outcome measure. So what did we do with the eye tracking? We looked at a few different things. We were particularly interested in, so there's a lot of eye tracking work in this sort of field of social attention in autistic groups. And so, you know, we wanted to see, could we tap and could we look at some of these autistic traits, measure them objectively, and maybe look at, so, you know, it can also give us some idea about mechanism and things. So we did a few different tasks. The first one is, well, there's three of them. The first one was this kind of social, the one on the right of the screen, which is, you know, you show them two scenes, one with a social stimulus, usually a person, or it is a person who's in it, and a non-social scene where there's no person in it. And you just look at how long people look at one versus the other. You show an array of pictures, and you look at how long people look at the face, which, you know, most people will orient towards the face and be more interested in the face than they are in the other things. And then in faces themselves, people typically are more interested in the eyes, and then to a lesser extent the nose and the mouth, feature parts of the face, but particularly the eyes, than they are in other parts of the face. So what we're doing is just, we're looking at where people attend to, you know, what people's eyes are drawn to, what they're looking at. And we're also, I should say, it's looking at relative time. So our SYNGAP kids do not look at the screen as much as the typically developing controls, because they are more distractible. So they would be looking away. But what we can do is look at the relative amount of time that each person, you know, so that somebody is looking at the whole screen, and we look at the proportion of that time that they are looking at the eyes, say, for example. So the first one, the social versus non-social scene. What we see is that actually the SYNGAP group performed pretty much the same as our typically developing controls. They were more interested in the social scene than the non-social scene. So more interested in looking at scenes with people in them than scenes without people in them. In the same way, exactly the same kind of pattern that you see in the controls. So on the left of this group is the social scene, SYNGAP and controls, and the right, non-social scene, SYNGAP and controls. And there's no differences between the the Syngap group and the controls here, and both tend to look at the social scene more. So they're interested in these kind of social scenes more generally and things with people in them. It gets a bit more interesting when we look at these, what folks look at in these pop-out tasks, as we call them. So there's lots of things on the graph there, but the interesting things we find is that the Syngap group look less at faces. They fixate less on the face than they do on the other items. They also look a bit less at the phone. It's a very old phone in this picture. I don't know whether that is because people are less interested, or whether the controls were looking at it thinking, what on earth is that? But you know, they were particularly less interested. I mean, the Syngap group showed less inclination to look towards the face than the controls did. And that's interesting because that's in a bit of contrast to the previous one, where actually both looked equally at the social scene. And then this gets more marked when we get to this more intense social stimulus. This is a kind of zoomed in picture of someone's face. And this is the one on the left is what the child sees, and then the one on the right is how we define non-feature parts of the face, which is the white oval. Then the red is how much people look at the eye region, purple the nose, and the blue is the mouth. And broadly, what we see is that kids with Syngap look less at the face. They're more looking at other parts of the picture. And they look less at the eyes. When they are looking at the face, they are looking at other parts of the face rather than the eyes. So even when the proportional looking time for the eyes is actually compared to the whole face. So when they're looking, they don't want to look at the face, and when they look at faces, they particularly don't want to look at the eyes. Whereas the controls show that bias towards eyes and towards faces. We just don't see that bias for the Syngap. So when we take these kind of social attention things together, we do see social motivation, and families will tell us that their kids are pretty social at times and want to be social. But they look when you get these more intense social stimuli, which is this is how we're interpreting this. That, those social scenes, the people in them aren't usually looking at the camera. There's lots of other things going on. They're not an intense social stimulus. But as they get more intense, you start to introduce faces that people have to look at, and then eyes and so forth. That becomes more difficult, certainly less appealing than it is for control. And it might be that it's actually stressful, that there's a kind of aversive nature of these kind of more intense social stimuli, which is something we see in some other conditions that, for whatever reason, that these more intense things can be quite aversive or cause a degree of stress. The other thing that's interesting and good about this is that, the eye tracking does seem to be able to objectively quantify some social differences. I didn't mention it there. I thought I had a graph with it actually, but the difference in time of looking at faces was much less, or was associated, children who are more autistic showed the greater aversion to looking at faces. When we looked at comparing these kind of eye tracking findings to our measures of autism, we found that they related. So they are indexing, these eye tracking findings seem to be objectively indexing these sort of autistic type traits, or social communication part of the autistic traits in our kids with Syngap1. The other thing we looked at eye tracking, and I will just mention this quite briefly because I'm a bit aware of time, was a thing called inspection time. The reason for looking at this is that in typical people, the quicker you find an odd object in an array or a group of other objects relates to your overall IQ, basically. This we thought might be an interesting way of actually trying to pick out, well, how could we index IQ or objectively using our eye tracking. So what we ask children to do is, we don't ask them to do this. We show them a picture, put up a nice attractive stimulus in the center, some little cartoon character or something like that attracts people's eyes to the center. Then we display this array of shapes, which are letters here, around. All of them the same apart from one. These circles aren't there, I should say. So these circles aren't actually part. And you look at how quickly people find the odd one out, and you look at how often they look at the odd one out. And mostly typical people will look at this, relatively quickly and will fixate on it more than the others. Importantly, for this to be able to do it, you have to look at the center. And there was 6 of our SYNGAP kids just didn't look at the nice, attractive thing in the center of the trial. So we had to exclude some people from the analysis. And what we find that of the trials, number of trials that people actually do, the kids with SYNGAP1 are less likely to fixate. They're less likely to look at this odd one out. So we only show this for a very short amount of time. And typically developing controls are more likely to, locate and find the odd one out. The speed at which they find the odd one out, the ones who manage it, it's a bit slower in our SYNGAP group. It's not significantly slower, but it's a bit slower. But we do see a relationship between between nonverbal IQ and adaptive behavior. We do see a relationship between these measures and the speed at which people fixate. So those people who have got a higher nonverbal IQ or a higher adaptive behavior within our SYNGAP group find this odd one out object quicker. So it suggests to us that it might be an objective way of kind of indexing cognitive abilities, which might be helpful or suitable for clinical trials. Okay. So I'm hoping you're all still with me. I'm hoping people haven't turned off from all of the stuff. I'm too much data. This is the last set of things I want to talk about, which is sensory processing in the last few minutes. We knew from talking to families, we knew from seeing children that there are sensory processing differences in SYNGAP1. And we used this measure called the sensory profile. And it's a good measure because what it does is it allows you to look across different senses. So hearing, vision, touch, movement, body position, and a mouth sensation domain. So it gives you scores across all of these. It allows you to tap sensory behaviors, which I'm not going to talk about, but some of the stress type behaviors that might result from sensory things. I'm not going to talk about here because we've not really looked at those in great detail yet. And then it allows you to kind of, look at these scores within this theoretical model of sensory behavior and sensory sensitivity. Basically, this has an idea that there is hypersensitivity and hyposensitivity. If you look up and down this arrow here. And then that's affected by how much people actively try and self-regulate their sensory system. So how much they use their behavior to regulate their sensory input, and how much they self-regulate using more passive measures. And then it gives you these scores in different domains. So somebody who is hypersensitive and actively self-regulating, they're hypersensitive to stimulus stuff. They will move, they'll avoid sensory stimuli. They'll try and reduce it by kind of withdrawing and hiding away, or they'll be very sensory sensitive if they passive self-regulate. So you'll notice that they are, particularly reacting to certain sensory stimuli. Somebody who's hyposensitive, if they're active, they might seek out sensation. They try and increase their sensory system's responses. Whereas if they're passively self-regulating, they just don't notice sensory things. So they just don't seem to register them. You don't really need to understand the kind of whole model to sort of think about the results. But I guess the key thing here is that we find this contains data from Fragile X syndrome, I should say, as well as SYNGAP1, so are these slides. But what we found was our SYNGAP1 groups show clearly, high levels of sensory sensitivities across the auditory domain, less obvious across the visual domain, but certainly with auditory, touch, movement, body position, and oral sensations. There is differences in sensory sensitivity. And this is both high sensitivity. If we look at these different domains, both hypo and hyper sensitivity. So both, seemingly low registration. So this is one of those classic things that people have talked about, not picking up pain stimuli, not seeing to respond to pain. That would be something that would come in here. This hyposensitivity. Sensory seeking is associated with like seeming to enjoy things. So the kind of stuff families talk about is like wanting to touch water and so forth. And that's meant, that could be an example of someone who's hyposensitive, but they're seeking stimuli. And then this hypersensitivity, avoiding, appearing sensitive, getting very distressed by various sensory stimuli. all of them in our group, we saw these differences in SYNGAP1 across, and they're similar to those that we see in Fragile X. And Fragile X is another condition we work in, where actually these are very well established, these sensory differences. One of the kind of core parts of Fragile X is these sensory differences that we see. We see similar levels in children with SYNGAP1. And then what's important to remember as well is that the same child can be both hyper and hyposensitive. So that sounds a bit contradictory. How can you be hypo and hypersensitive? But they can. This is a dysregulation of the sensory system rather than an oversensitivity or an undersensitivity. So it is, and that's what this graph on the bottom left here shows, that children who are more hypersensitive are also likely to be more hyposensitive. And that's a really strong and consistent relationship. So you can't think of just one or the other. But hypersensitivity is the thing which is most associated with these difficult behaviors that I've talked about earlier. These sort of aggressive or distress type behaviors are most associated with this people's levels of sensory hypersensitivity, which suggests that it's particularly important to try and understand those in terms of when you're thinking about what might be causing sort of behavioral difficulty. We then went on to have a look at sensory processing using EEG. So seeing if we could again quantify these sensory processing differences and look at maybe what might be some of the things that underlie them. We did this using what's called an auditory oddball paradigm. So basically you have this EEG cap on and we play lots of tones, 600 tones, beeps basically. And then some of them are a certain frequency and some are a higher frequency. So it's really like beep, 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 beep. And the reason for doing that is it allows you to look at how sensitive somebody's brain is to their auditory system is to these tones, but also what's different whenever they're hearing the same tone over and over and hearing a different tone. And that's what the graph on the bottom right sort of shows. This is the kind of response, electrical activity in the brain that you see to these repeated tones. And then the oddball, so the odd one out, the higher tone, you get a slightly different one. And we're interested in that because this difference is a thing called mismatch negativity. And that's interesting to us because it's possible it indexes a few different things, but one of the main ones is it's thinking about a thing called prediction coding or prediction error. So your brain ability to look at these, our brains rely on basically being able to predict things about the world. And what it's predicting when it's hearing the beep, 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 the repeated tone, is that that's what I am going to hear. And then when it hears something different, it goes, oh, something different has happened. So it's having to change its predictions. It's having to adapt in terms of how it is working to a change in the sensory environment. And that's the thing which is called mismatch negativity. And what we see basically in the children with SYNGAP is that there are differences in the mismatch negativity. So it's a bit hard to see in this graph, but the green or the blue is the typical developing control, just listening to the repeated tones. The green line is then when they hear the standard or the oddball tone. So you get a difference occurring at this point, 200 milliseconds after the stimulus onset. SYNGAP group are the black and the red, and you don't see a difference at this point. They're not reacting differently to the oddball tone as they are to the standard tone. And that suggests that this predictive coding mechanism in the brain it's not processing the sensory stimuli in the same way. And then what's really interesting about this is that when we take this EEG derived measure and then we correlate it to sensory hypersensitivity measures, it correlates quite strongly. So the more hypersensitive kids are the ones who show the biggest difference from controls in their mismatch negativity. So basically it suggests that this lack of the difference in the way that the brain is processing this oddball stimulus is in some way associated with the degree of hypersensitivity that people show. And it's also really strongly associated, albeit numbers are pretty small at this point, that we're looking at, with the levels of difficult behaviors. That this predictive coding difference in the brain, this mismatch negativity, is associated with differences in sensory processing and increases in these aggressive or agitated sort of behaviors. So pulling that bit together, where I've gone over my time, that, you know, we see high levels of sensory hypo and hypersensitivity, and they can occur in the same children across different senses. They're probably really important for behavioral challenges, particularly the hypersensitivity. You know, and it's one of those things that, you know, we increasingly think about in clinical practice. Is getting, you know, it's usually an occupational therapist who would be able to look, measure sensory processing differences and things, and then try and give some strategies to try and manage them. It's not like you can get rid of these things, but how you might be able to manage some of these sensory processing things by optimizing the sensory environment to try and reduce these levels of difficulty. Then we have some idea, and it's preliminary data in small numbers, so we actually need to analyze this more to check that it holds, and that this sensory hypersensitivity might relate to how differences in the way the sensory processing system kind of codes, predicts stimuli that are coming, and then alters itself to new stimuli, and that we might be able to, and we can capture that using EEG, and that in itself might again be a biomarker, another way of tracking things in trials. Okay, this is my second last slide, promise. What are we doing now? So the biggest single thing that we're doing related to these data is that we are going to try and follow up lots of people who've taken part in this. As I said before, we need to do longitudinal studies to look at how behavior changes over time. To try and look at, you know, to help people understand, you know, what they might expect, what are the things that we might need to look out for. And B, because having this, what we call natural history type information, gives us if we're going to have interventions and trials and treatments and things, you need to have some idea about how the condition changes and progresses. And this is one way of looking at it. You know, there are other ways, and I know that SRF have funded the citizen data to look at natural history, and I think that's really good. But this is a way of trying to look at it with some of these quantitative measures. We're going to do this. We're starting it in about 2 weeks' time. Sydney, who's the person on the right of this, is going to be running this study. We're going to repeat the measures, and we're going to recruit new people, and we're going to be aiming to follow up people over time in this. We're particularly also, though, going to focus on some of these aggressive and difficult behaviors, because that's one of the things that causes a lot of distress for people. So looking at sort of what are the causes, what are the markers, what might be things that might be able to be targeted, I guess, for potential treatments, and things around these sorts of behaviors. And so she's also going to look at things like autonomic arousal, nervous system arousal, using EEG. And so that I'm going to start seeing the first groups of people in a few weeks' time out at a family day. And then Lindsay is expanding her sleep study. Hopefully, she's awaiting some funding decisions around that. But we are. The hope is that she will be able to go and to look in more detail at some of these measures. And that is me. Just thanks to everyone who's been involved in the research, all of the families who've been involved. Thanks to the Syngap Research Fund, who've been a great supporter of us, as well as Syngap UK and Syngap Foundation. And then this is Damien, who did a lot of the work. Aisling is over here in this picture. This is our kind of family day. You know, and this is us showing our stripes for Rare Disease Day. I should say we don't normally have lab like that. But yeah, most of all, thanks to the people who've taken part, because we can't do clinical research without people taking part. Thank you.
That was epic. I got a lot.
Very long.
No, it's good, and it's good you have COVID. I'm sorry you have COVID. I hope you get well. But it's good you have COVID because you slowed down enough that we could actually absorb it. I think if you'd been at usual clock speed, we would have lost us. But there's a number of good questions in the chat, but I want to abuse my position and just go through my questions quickly, because I fear that what you've just done in an hour really should have been a day of a tremendous amount of work. before I jump in though, some of these things said Wright et al. Submitted, some of them didn't. So I'm assuming that the Wright et al. Submitted is coming to publication, whereas the ones that didn't say that on the bottom of the slide are still works in progress and we can look forward to a future publication, right?
Yes, very much so.
I want to make some broad observations about what I said at the beginning before we do all the cool sensory stuff. You referred to autism and ADHD, and I think even in the US, I find that Syngapians, noting the eye tracking and the preference for faces, right? The average younger Syngapian tends to walk into a clinician's room for an autism assessment and does what they've been trained to do since they were 2. They lock eyes with the clinician and they smile beautifully. And the clinician says, not autistic, right? They then get through the assessment, go home and break everything. So, even in the US, I'll observe that someone needs to hit mute. I'll observe that it's hard for our kids to get an autism diagnosis. And I guess one question for you is do you get the sense that it is also hard, if not harder, in the UK to do the same? So that's question one, is how hard is it to get that diagnosis in the UK? I mean, question two, I also point out that until recently, and it is changing, because I'm starting to see 2-year-olds. In the US, you got a SYNGAP diagnosis because you were having eyelid myoclonia or worse seizures, right? Whereas in the UK, because of the NHS and the Triple D, you guys have been sequencing people with ID. So it follows, doesn't it, that your percentage of patients with epilepsy is lower than we would expect from the literature, which is not all from the UK. And does it suggest then that SYNGAP it supports the hypothesis that there's a lot more Syngapians out there, we're not finding them. And maybe there's a lot of them who are just not seizing but are delayed. So that's question two. Related to the epilepsy, this is a slow moving phenotype, right? So some of our very severe cases start having drops in the early years. But generally, I would say it's absence, eyelid myoclonia, progression over time, drops. So as you're observing the behavior and all these things getting worse over time, before we even talk about adolescence and hormones, would it make sense that as seizure frequency increases, and if early seizures are absence and missed and therefore untreated, just the noise in the brain gets greater, and then it's a plausible hypothesis that would explain a lot of these things we see getting worse over time. Let me stop with those three.
Okay, cool. I'll try and remember them all. My brain is not functioning as well as normal. So the first one is about the autism diagnosis. That yes, I think it can be challenging in the UK in the same way that you described as in the US. I think it relates to. I mean, I think there's a few things. I mean, there's the kind of, as you say, if you've got people who are, a socially motivated individual, in the way that we describe, sometimes there is this idea, well, how autistic are you, etc., and that then pushes people away from the diagnosis. I think as well, though, there is a tendency, and it's hard to know what, it gets very difficult when people have got quite marked communication difficulties and impairments in their kind of IQ and intellectual abilities. There is a tendency has been a tendency not necessarily to give an additional autism diagnosis, to say, well, look, it's what you have is SYNGAP, what you have is moderate to severe intellectual disability, what you have is, we see this in Fragile X and other things, and actually, your social communication and so forth is what we might expect for somebody with this level of intellectual kind of impairment. And I think that I don't think that's always going to be very hard to tease out. That's in part diagnostic trends and habits. I think it's maybe a bit more likely that people get autism diagnoses now with these difficulties than would have been the case 20, 30, 40 years ago. And it's in part this kind of odd and almost unsolvable thing to say, well, what exactly is autism in this sort of context? My view is very much, if it adds to the understanding of a child and the clinical picture, these sort of, particularly if you think about, we think autism, yes, social communication differences, but repetitive behavior, sensory issues, and so forth. Like, those are things which are important for people to be aware of, and to highlight. And I think having an autism diagnosis helps in terms of highlighting those things. So that's, I mean, in a very pragmatic way, I think it's helpful.
What about
yes. So the epilepsy, yes, these cohorts are derived primarily from research. This research cohorts that were based around developmental differences occurring earlier on. So they're not. So you're right. If you look at a bunch of people with epilepsy, and you identify SYNGAP, you're going to come up with the idea that all SYNGAPians have epilepsy, and that's, whereas if you look at a bunch of people with intellectual disability, some of whom have epilepsy and some don't, and you identify SYNGAP, you come up with a different idea. So it is, I think, where you select from is, gives this thing. Our findings are definitely consistent with the idea that there is people out there who either don't have epilepsy or you have such mild seizures or whatever that they're not noticeable. You know what I mean? That this more severe phenotype is not there. But I would say, like I say, 60% of our group had seizures, so 40% didn't have noticeable epilepsy. So yeah, it does.
So every SYNGAP parent will smart when you say mild phenotype, because a mild subclinical storm in the head times 100 in a day will still mess up your day, right? Especially if it's every day. But
sorry, I shouldn't use it.
No, I'm not being difficult. Yeah. I'm just advocating.
No, I
know.
My job.
But I want to push forward a little bit, because there's a lot of great questions in the chat, but you've just triggered that webinar was epic, and I will be sending it to every industry partner and every clinician, because you touched the tops of so many icebergs, and I just wish you had a bigger N on everything. Not a criticism, just a supportive comment. So as the N grows, though, I think a fair question will be, can you break that data down by medication and phenotype and genotype, pardon me, right? Was it a missense? Was it a protein truncating? So I think even if you don't have that, being able to share the genotype of the subjects might be interesting, even if in a table. And I want to talk about drug for a second. I just get anecdotal reach outs from parents, right? But parents call me when things are blowing up generally. And the first question I ask is, what drugs is the kid on? I will observe that in the US, kids generally are on more drug. And what I say, I'm not a doctor, I don't give medical advice, but I do say, I find that generally SYNGAPians do better when they are on anti-impulsivity drugs like guanfacine and anti-anxiety drugs. Ask your clinician for those. In the US, anecdotally, patients go and have those meetings. In the UK, patients tell me, no, I'm on one epilepsy drug and my doctor said I'm good. So I just wonder if, so there's two questions there. One, would it be reasonable to assume that this cohort is generally on less meds than, say, a US cohort? And two, how would we, globally speaking, effectively suggest to the clinical community that, hey, your kid's got SYNGAP. Maybe in addition to ASMs, we should be thinking about anti-anxiety and other behavioral meds, not to make this kid more manageable, but to reduce the suffering of this child who is going through a world that is, I think you're exactly right. My personal hypothesis, having done nothing but think about SYNGAP for 5 years, is the world is, and I learned this from JR, who's asked a lot of good questions that we should get to. The world is incredibly anxiety inducing for our kids. And I think that triggers both seizures and behaviors.
Yeah, so you're right. They will be on less medicines than those in the US. I mean, there's both good and bad things to being on. We don't want over medication, but we don't want under medication. And I think that there is as I think you say, and I agree with you, and I touched on my talk, I do have some concerns that people would benefit from medicines that maybe they're not having, and not, often for things like agitation, people end up getting prescribed antipsychotic medicines and things in the, and stuff. And, whereas actually it is possible that medicines like guanfacine, as you say, or anti-anxiety medicines, which have probably a better side effect kind of profile, are likely, may potentially have effect, How do we change that? Partly, I think getting information out there and saying, these are behavioral problems which are pretty marked and cause issues, and that even saying 7% of the kids that we saw were diagnosed with ADHD, whereas they all showed high levels of hyperactivity and impulsivity and things. That is a message that we need to get out there from this kind of study. I'd love to do, I think we need to try and start, there's tons of trials that need done, isn't there? But there's trials that need done targeting things that, hopefully we're going to hit stuff like gene therapy and ASOs and these things. But there's also questions about, well, what are the medicines that might be helpful? Guanfacine is a really interesting one. I would, there are colleagues of mine who are looking at it in some of the laboratory models and things like that as well, to see what are the medicines that we have that we should be suggesting might be particularly helpful for these groups. So, yeah.
I want to go to eye tracking. I'm trying to move really, really fast.
Mm-hmm.
I will observe, by the way, there is, as the phenotype broadens, there are certain subphenotypes of our kids, the C33 DELs. Those kids are a step function higher on ID, right? So if you have one of them in your cohort, it's worth calling that out. There's also another subcohort, my son included, who is oddly verbal. And what I've learned from Tony, the verbal, there's some kids out there on trazodone. Tony on trazodone is so hungover the next day, he can't complete a thought. Kids who are nonverbal, you wouldn't catch that. But just to continue with my guanfacine commercial, only when we put Tony on guanfacine ER did he sleep through the night. So it was an impulsivity drug that actually ended up working for sleep, right? So the holistic view here that you're forming is beautiful. I want to just make a point on sleep and then go to eye tracking. Sleep, you referenced the Smith-Hicks paper, very good paper. I agree with it completely. They don't cite in that paper Sullivan 2020. And maybe I've got my years mixed up, but I think they could have. And in Sullivan 2020, what you see in those human EEGs and mouse EEGs was that seizures cluster at sleep, wake, and wake sleep. And I've always wondered if that is one of the reasons for sleep resistance, because these kids know they're heading into a seizure storm. But I want to go to eye tracking. So Tom, you guys are doing everything you said makes sense. Tom Frazier is also doing a tremendous amount of eye tracking work, and I hope you guys are in touch. And if you're not, we're funding it, and I want to connect you because you guys are barking up all the same trees. Okay, the only other thing I want to ask you, and again, that was just a beautiful review of so many things we could use as biomarkers. I didn't hear you talk about GI, and I'm wondering if in the forward-looking stuff, you may want to. Are you planning on collecting any data on that?
On GI, like the gut?
Well, I don't want to head you down the gut-brain axis and microbiome and all that stuff, but the plot I'd love to see is behaviors against how often does your kid poop every week? Just to be blunt.
Yeah, it's interesting. So we're not, interesting enough, Aisling, who was the RA, who was working in this study, is now doing a PhD about the microbiome in premature birth and things like that. So, it's an area which is really of increasing interest. We're not collecting any data around that at the moment, but there is definitely GI issues, that I don't need to tell you guys that occur.
Again, like we're just a civilian observation. When kids stop pooping, seizures and behaviors go up. And when people go ahead.
I was going to say, I work part of my clinical job, which is just a more general NHS kind of. I work in an inpatient ward for people, adults with intellectual disability. And one of the first things that we think about when we see behaviors change is have they opened their bowels? And it's one of those things that gets reported. And and there's lots of different things there, both about just discomfort and so forth, but also there is broader gut-brain axis and stuff questions. Yeah.
All right, I'll hand it over to the Q&A. You've got 11 more questions. Good luck with this. I'll call out the people I know. So JR has a few asking about the ADHD, that second question. Do you want to read it? Because it's pretty long.
I'm very interested in the ADHD associations and looking more closely at threads of symptoms. Hyperfocus and inability to switch attention is one side, but another issue is the reward pathway, a strange addiction cycle with preferred activities and objects combined with access. What starts as something interesting can become a favorite, then a requirement, then becomes a painful while screaming for it. And I wonder if the addiction type escalating behaviors are based on a reward pathway, and if that is part of ADHD. So yeah, I mean, that's a really interesting kind of formulation of some of these kind of difficulties. That addiction cycle is something that you see in children who with ADHD more broadly sometimes, these sort of whatever these dopamine hits that people get, and that there is a difference in the way that the brain experiences reward and reacts to things. And then stimulant medicines, which are the ones that are used mainly for ADHD, different than guanfacine, but guanfacine probably also affects dopamine a bit, but stimulants particularly affect dopamine and likely do affect some of these kind of reward pathways and things. So, it is, you could sit and do a PhD, a number of things based on that question. But broadly speaking, that yes, it is possible that these kind of reward pathways function differently, as we think they do in ADHD, and as we are interested in autism and other things too, and that need for dopamine almost is what driving that requirement and that painfulness and things that they're talking about.
These are all good. Do you want to just go down the list?
Just to keep going down them. Someone's asked if we look at behavior difficulties between males and females. We didn't find much in the way of differences between males and females in this group. That's not to say you wouldn't in a larger group. You see the same kind of patterns of behavior basically in our the cohort that we had. I think sometimes, and I'm just thinking clinically more broadly, as they get older, males are bigger, hormones change, some of the behavioral consequences of these behavioral difficulties become greater for people. But I know lots of people with female children who struggle with a lot of the same kind of difficulties. And like I say, we didn't see any clear male-female difference there. Okay, someone's asked, how early can the diagnosis be made for autism, ADHD, and anxiety? Earlier than the average population, and how to get around the atypical autism of the Syngapians that are more social. We kind of touched a bit on the last part of that, and I think it is highlighting the difficulties that you guys experience with this resistance to change, with these repetitive behaviors. Labeling what is typical social behavior for someone with a global intellectual impairment is really hard. Massive struggle with sensory differences, massive struggle with change, resistance, routine, etc. Those are things which are a bit more quantifiable and can be pointed out. How early the diagnosis is difficult. It's probably actually not that much. Other difficulties are likely to be noticed first, and that can lead you into diagnostic pathways earlier, but it's not like you can pick these things out, I guess, necessarily earlier. Anxiety is a really tough one as well, because so much of anxiety, relates to how you express it. And if it's expressed differently, because people don't communicate, they don't say, I'm stressed, I'm worried, they act differently. Picking out what is anxiety and what is not is difficult to say. I think a lot of these distressed behaviors are associated with stress, and stress is a precursor to kind of anxiety. Anxiety is a kind of higher construct, but it's stress and distress, I think.
Yeah.
Someone's asked about hormones in adolescence. Do some of the behavioral difficulties in adolescence, could they come, are they hormone related? Does it plateau at a certain age and maybe wanes, or does it continue to increase in severity throughout adults? I don't know the answer to someone. They've said, is there an age that families feel, okay, we made it to the worst and it should level out or get better from here? I would say that we don't have studies. We need to know more about the adults. We just do need to know more, and more adults will be getting diagnosed as sequencing widens out, but it's all, children are the people who get diagnosed first. When these new tests run out, they're prioritized for the younger populations, but there'll be lots of Syngap adults out there. I would say that, adolescence, is a time of difficulty for all parents, where changes in behavior as families grow, people change, resistance to hormones, sexual feelings, and behavior, very difficult for people to understand, even harder for someone, a Syngapian, to understand and process. Our general experience is that there is a sense, that you can make it through the worst and it should level out or get better, as you say. We don't know that for sure, but that things, are likely to be adolescents are more emotionally labile, they're more volatile, and they've got a whole lot going on. And that should improve a bit as people move into adulthood. Not for everybody. If I think of other conditions that we work in, might be different, like things like Fragile X and stuff. Adults are much calmer than the kids. Kids have much more marked ADHD type features and things, and the anxieties are still there, but they're not as prominent, and the ADHD stuff does settle down a bit. So yeah, we need to look at it more, but I would say that yes, and I hope you don't. That was one of my worries about presenting this. I don't want people to go away and thinking, oh goodness, it's going to get harder and harder. That's not what I'm trying to say. It's to say, what can we think about? What do we need to look out for? And maybe is there things that we can do that might make adolescence easier, is the importance of that. It also allows you to go to clinicians and say, look, things are getting more difficult. We know this happens in this population. Please take us seriously. Don't just dismiss us. I think that's the other thing.
I don't want to break your flow, but
No.
I want to underline what you just said. I for the first few years, we were very nice and gentle with the newly diagnosed parents, and I've taken the gloves off because just 10 seconds. I had a newly diagnosed family who was in the process of moving to a new state that was very hot and about to buy a new house. And they're holding a 2-year-old and they're like, my baby doesn't seize yet. Maybe we're not going to seize. And I'm like, hate to be a jerk, but pretty good odds your kid's going to seize. I'm looking at a protein truncating variant here. And I think it's important that families walk in 2 points. One, clear-eyed. These kids will get more complicated and become bigger for the next 15 years. And then at adulthood, I think this is what's sort of behind explicitly in the end of Jackie's question. They do tend to level out. So I don't think it's a message of despair. I think it's a message of hope. Like, hang on, love this kid, get them to adulthood. They're going to be okay. Look at Karen. She's 65. This is not a terminal disease. And I think your point is probably the most important. Walk in waving whatever publication, hopefully your next one, is needed, and insist that these clinicians find the time or give you another appointment, take this seriously, and help us reduce the suffering of and increase the manageability of these patients, because they are complicated. And I think Syngap enjoys a huge wealth of cool science, but we're really suffering from a dearth of clinical work. And I think your work and Andrade's work and Connie's work and all the other good stuff is helping us close that gap. But it's desperately needed because the families are struggling mightily. Okay, buckle up. You got another JR question. It's PhD level.
So I think it's more of a comment. It's about how the idea of older people having more severe phenotypes seems like a reasonable observation based on who gets the genetic tests and when they're likely to get them. So that's in this cohort, the older kids, where people who ended up getting exome sequenced early, and that's not offered to folks, that's maybe more likely to be done to people with, more inverted commas, severe kind of phenotypes and things. I think as we expand exome sequencing, we will find more SYNGAP variants in people with milder forms, milder, I'll do my inverted commas again, but of intellectual disability and things. That doesn't mean they won't have other difficulties, but I think we will see a kind of expansion of this. I think the people who get diagnosed are the ones who are ending up in research studies, the ones who've got pretty severe epilepsy, and sometimes in people who, for whatever reason, have managed to navigate their way through a system and to get these things. Exome sequencing will increase, and we will see more, and I think adult sequencing will tell us a lot and hopefully answer and reassure probably some of the questions that people have about what's the future going to be like. As you say, Karen is 60, well, she was 64, now 65. As we can describe people, that's gonna be helpful. Oh, for some reason or other, one of the smoke alarms has gone off in my house. I don't know if that's coming through.
It is
strongly. It is
not too strong. Do you need to make sure there's no fire?
there's other people in the house, so I will get told. I
Okay.
suspect dinner is being cooked here at 6:30.
Sorry to push you, but next question is
Yep.
nonverbal IQ.
So it is measured with a test that's lighter. Basically, it's getting people to match. They're looking for specific pictures. You show them things and you're indicating to them to look at things. It's about recognizing shapes, finding things in larger pictures. And you use a lot of gesturing and things. It still involves communication, but it is less reliant on that than kind of verbal IQ. But it's a lot of matching, sequencing, things like that.
I wonder if those scores would go up if you gave them more time. Because
yeah, possibly.
I mean, Tony, I mean, I can only speak for my own case, but my kid will give you the right answer if you give him an uncomfortable amount of time to respond. But it's always the processing speed is just low. I have literally said to therapists in my house, you would be more effective if you came to work stoned. Like, please go get stoned and then come work with my son. If you were slower, you would be more successful with my kid. And they think I'm kidding, but I'm not. Anyway, next question, hypo.
Doing different domains are changeable. Hyper and hyposensitivity going together, do you mean in different domains or changeable in the same environmental situation? So even within the same domain, I don't think they are particularly changeable. But you guys may say different in terms of if somebody is hypersensitive to something, to noise, it's unlikely that in other situations they would be hyposensitive to loud noises, you know. But within a domain, they might be hypo and hypersensitive. So certain loud noises, they might be hyper and they might be hypo to them. Certain touches or feel of things, people might be hyper to the touch of something, but hyposensitive to pain. So it's a really complicated, and it speaks to, and I don't think we really understand this very well. How can you be both hyper neurologically, how can you be hypersensitive to touch and maybe hyposensitive in other things? So it can be, it can be within the same sensory domain, and it can be within it can be the same across different situations. Well, that makes sense. We need to know more about I think sensory stuff is so important and has been so little looked at over the years in this sort of field. And it's only really in the last 5-10 years we've started becoming really aware that actually these things are key to behavior, key to people's ability to process the world, and key to anxiety and things like that as well. If we can tap into and work out ways to help some of these sensory things, then that I think could go a long way towards, improving people's quality of life, which is ultimately important.
But let me add to that list. Temperature control. Not all, but many of our kids tend not to sweat very much. And I think that is the tip of another iceberg, like thermal self-regulation, right? If I look back over, summers are hard for our kids. Summers are hard for our kids because there's no school, everything changes, blah, blah, blah. Summers are also hotter. And if I look back over some particularly challenging behavioral incidents, it's generally on hot days. And I think our kids are uncomfortable, but their body is not managing it, in part maybe because they're not doing self-regulation on the temperature side. That's something we cannot find in the literature, but anecdotally is popping up like crazy. Okay, Aaron's throwing the Dahlman paper at you.
Yeah, I'm not a zebrafish scientist, but I have some familiarity with some of these things. I work a lot with people who do most of the work in Edinburgh, I think you guys probably know, is done using mice and rat models and things. But this idea, I mean, the quote you have there, that this causal for hyperactivity associated with elevated arousal, especially pronounced in low arousal environments. I don't know that we have, it fits with the idea that people are in some way trying to regulate something by some of their activity, that arousal levels, if somebody is in a low arousal environment, they might then get more hyperactive. I have to say, that then feeds so into ideas about just distraction and other things. And ideally what we tend to do is reduce arousal if somebody is very aroused. We try and bring them down. So I don't know how much that would hold across in humans. One of the big problems with any clinical kind of research is a lot of what we do is very artificial in a way. We do these things in laboratories. You know, we see people. We're not actually seeing people in a home environment and across multiple different environments. You know, so yes, I don't know is the simple answer, I suppose, overall. I'm very interested more broadly in translation between our laboratory models and people, and as like that's going to be key going forward. And I've not touched on that, but we are doing some work. We are a number of pieces of work actually to look at translation between some of the rat models of SYNGAP1 in Edinburgh and some of our clinical findings, which have not presented today. But, okay, the next one is the oddball tone test. I wonder if you could try a more complex test where one listens to a simple song with a strong constant beat and then alter part of it in an unexpected way, but keep the same rhythm. This is because of the background wave slowing. I wondered if having a beat is helpful for listening. I'm going to say possibly, I don't know. I think I would need to speak to somebody, you know, perhaps Damien, who did some of this work, but with someone who's a more auditory processing scientist. You know, you know, I don't know about the effects of kind of the rhythm and what that effect has on. All of these are played in the same rhythm that we have. You know, it is literally boop, 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 boop. You know, and so I don't. In it, it's a very artificial thing. Having a more complex thing with a song and a beat. Yes, I don't know. Don't know. You end up with so many variables to try and control. You know, that's the thing that starts becoming quite difficult. Okay. And then, I think the last question is, why is an additional autism diagnosis beneficial? In the UK, sensory processing difficulties and the consequences are much to overlook. So going in with a SYNGAP1 diagnosis gives us more opportunities to push practitioners to understand this part of the puzzle better and then get those OT-SI referrals. Yeah. So I think an autism diagnosis can be beneficial. It's not that beneficial in terms of things like understanding social communication. I think it is beneficial in highlighting sensory issues. I think if you go with a SYNGAP diagnosis, to most clinicians, they're not going to know anything about it. And at best, they might go away and look at something, you know, they should go, but they might go and look something up on the internet. If getting an additional autism diagnosis, it does highlight this sort of routine, sameness, sensory kind of things, you know, which I think that is what is beneficial. I mean, there's always caveats to these things. What you don't want is people going like, oh yeah, cool, they have autism, so we just need to think of this with an autism lens, you know, because that's, and I see that in other conditions, it's not quite the same as whatever typical autism is, you know, but I think it's a label that society is familiar with, and that care workers and things, they will think more about sensory issues and think more about this kind of need for sameness and stuff with these kind of diagnoses.
I agree. I think you get the baggage and the opportunity with autism, right? You get the opportunity for more services, whatever, drugs, and then you get the baggage of, oh, it's just autism. Some people are I mean, it's such a big umbrella, whatever. In the US, it's essential for services. So it's a no-brainer. I tell people to go to their clinicians, give your child Red Bull, Snickers, take away their iPad, send them in, tell them to break things. Because otherwise they go in, they smile, and then they come home and then they break things. And that doesn't help anybody, right? Because that appointment took 6 months to get. Yeah, I'll get off that. So thank you so much for delaying your dinner and for giving us all your time and indulging all of my editorial comments. I really think your work is critical, and we're grateful.
Yeah, no, thank you very much. It's been really interesting to speak, and great questions and things like that. So, yeah, delighted to come and talk. And my house hasn't burned down, so that is a good thing.
That is a relief. Yeah.
Yeah.
Okay.
Excellent. Okay. Thank you very much.
Bye.
Thank you, Andy.
Bye-bye. Thanks. Cheers.